mistakes. I have a few observations about that quiz. One of the first, go ahead, Lourdes.
Good morning, sir. I have not done the quiz because when I was taking it, I had unforeseen technical issues and I sent you an email and I was wondering if I could take it in person.
All those people who have sent me email for whatever reason that they couldn't do the test, the quiz. If you had sent me an email, I will reschedule and make up for you guys. And I have that list. I have that list of names. So all I can tell you, be prepared, because when I say come to my office a certain day and a certain time, you need to show up. I will check your schedule, make sure that you don't have class during that time, so that you can show up to my office. All right, so all those who sent me an email justifying why they didn't do the quiz or why they couldn't finish for whatever reason, I will prepare and make up for you guys. Okay?
Thank you, sir.
Okay, you're welcome. Any other questions from those who sent me email? All right, so I was saying that I made a few observations from that quiz. One of the first observation is that you guys don't read your questions. Probably you see the question and assume that I'm asking something when in fact I'm asking something else. But you don't read. The second observation is that you do not proofread your answers. So, for example, one of the questions says, researcher studies partial linkage in two genes in roses, those responsible for flower color, as well as for flower arrangement. After carrying out the F2 generation, she obtained 1,500 parental phenotypes out of a total of 2,000 plants. Calculate the distance between these two genes on the chromosome. What was the percent of crossover frequencies that occurred in phase 1? So I gave you parental phenotypes, when in fact, to calculate the distance, what you need is recombinant phenotypes. So what you had to do is subtract the 1,500 from 2,000 to get the recombinant phenotypes. And so the recombinant phenotypes would be 500. So you're supposed to divide 500 over 2,000 times 100, and that will give you 25. So the distance between the two genes would be 25 map units, all right, or 25 centimorgans. That's the distance between the two genes. And then the percent frequency crossover would be 25%. All right, so obviously some people didn't read properly the information that was presented to you. All right, so you have to be extremely careful and read the questions. Like I said, the other observation is that students don't proofread their answers. The kind of grammatical errors in their English is not, I don't know how to say it. Some of the things that you guys write for your answer, you would think that it is a standard 3 student writing that. Some of it makes absolutely no sense. And when you talk about misspelt words and so on. So from this point forward, any evaluation that I give you and you have to write an answer, your answer can be as perfect can be. But if it has some grammatical errors and misspelled words, you will not get full points. I will take away points for English and grammar. All right? So make sure that whenever you're answering structured questions for me from this point forward, that you go back and proofread your answers. So So go back and proofread your answers and make sure that what you're writing there makes sense. Make sure that you can actually read it in proper English. And of course, there are several apps on computers that can help you in terms of spelling words properly. All right, so make sure that you guys proofread your answers before you submit. Okay. I noticed that there are only 38 people in the class, so out of a total of 52. So I would just take it that they're affected by the rains and the floods. All right. But if you notice what I did in that quiz, right? I said, for example, describe the example discussed in class. So you can go to Google all you want and get examples from Google, but I'll be asking for the ones discussed in class. So if you're not in class, then you won't be able to answer that part of the question. And so I will start editing my questions in a way whereby you will have to describe what we discuss in class. So it will be something like that. Use the example discussed in class and explain blah, blah, blah. If you're not in class and you're not paying attention, then I don't know how you'll get that right. But it will not be a question where you can go on Google and copy and paste. Okay, well, since you guys are asking, Indale wants to go over that question of finding the distance between chromosomes, and then Madison with the interkingdom one. Parental phenotypes is equal to 1,500. All right. Total plants equal 2,000. In our discussion previously, we said that for you to find the distance between the two genes, what you need to do is divide the total number of recombinants over the total number of plants. So if parental phenotypes is 1,500 and you have a total of 2,000, then obviously if you subtract 2,000 minus 1,500, that will give you 500. So the recombinants would be 500. So to find the distance would be 500 divided by 2,000, which is the total number of plants, times 100. And this will give you 25. 25 MAP units or 25 centimorgans. And that is the distance between the two genes on the chromosome. And then the percent frequency of crossover is the same figure, but in percent. 25% crossover frequencies took place in prophase 1. Is that understood, Indali? All right. Madison asked for the Interkingdom one. I had explained in class that there is a soil bacterium which is called Agrobacterium tumefaciens. This occurs naturally in the soil. It has the chromosomal DNA. Remember, this is a prokaryote. But it also has a TI plasmid. TI stands for tumor-inducing plasmid. Why? Because on this plasmid, there is a portion which is called the tDNA, transfer DNA. All right? So remember, this is the Agrobacterium tumefaciens, right? Which is found in soil. So this is something that occurs naturally. When this bacterium comes in contact with plant cells, for example, you're chopping around a citrus plant or a mango plant, and you accidentally injure the roots and you expose those cells, these bacteria that are in the soil come in contact with those cells. And what they will do is that they will transfer this portion of the tDNA, which contains some harmful genes, some genes that will cause tumor. So they transfer this portion, which is the tDNA, over to the plant cell, and that portion gets incorporated into the genome, right? They're into the genome of the plant cell. And when that happens, that plant starts to develop a tumor, all right? That tumor is actually called Krongard disease. So this is an example of interkingdom conjugation. Remember, conjugation is when you have cell-to-cell genetic material being transferred via cell-to-cell contact. So you have two cells coming together and you have transfer of genetic material. That is very common in prokaryotes, where you have two bacterial cells of different mating types, F plus and F minus, come together and they transfer genetic material. But I also mentioned in class that you can have interkingdom, whereby you can have a transfer of genetic material from one cell of one kingdom and a different cell from a different kingdom. In this particular case, we're talking about bacteria and the Plantae kingdom. All right, so I mentioned that genetic engineers use this particular attribute of the Agrobacterium tumefaciens, and they use it as a biological method of genetically modifying plants. So what they would do is they would remove that tDNA portion and replace it with a construct that has gene of interest. For example, you want to make that plant more resistant to a particular disease, or you want to make it, for example, the safflower that we mentioned, you want that plant to produce insulin for you. Then right where that tDNA is, you remove that tDNA portion and you replace it with those genes of interest, the gene that is responsible for insulin. And then you co-culture cells from the safflower plant along with that genetically modified Agrobacterium tumefaciens. And that will transfer those genes of interest to your plant cells. So that is a biological means of genetic transformation. Is that clear, Madison?
Yes, sir.
All right. I want to stress on another point, another observation. The question asked, explain conjugation in bacteria. Explain conjugation in bacteria. a lot of people answer that question with one sentence. You know what they write? It's when you have genetic material pass through cell-to-cell contact, full stop. If you recall that diagram that I had on conjugation, if you were to explain that entire diagram, you can't do that with one sentence. Because if you recall that diagram on conjugation, you have a case whereby the plasmid makes a copy of itself, all right, when these cells are in contact, and a copy of that plasmid go over to the F- cell. So all those details about conjugation and plasmid replication and so on, nothing like that was ever mentioned in your answers when it should have been. When you say I see explain, you can't do that with one sentence. You have to go in detail. And basically what you were supposed to do is explain that diagram that I presented there on conjugation, where you have two cells, the donor and the recipient, right? The F plus and the F minus coming together, they come in contact, sometimes with the use of bridge-like connections called pi-line. And then the plasmid makes a copy of itself, it replicates, and it passes that progeny plasmid over to the recipient cell. So at the end of that process, you will end up with two cells that would be F plus, because now that recipient cell has a copy of that plasmid. All right, so that's the kind of explanation I need when I say explain. So be careful when I say explain or discuss. That is different from if I say mention or list. If I say mention or list, it's just a mention, bam, 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 bam. No explanation is required. Is that clear? Any other questions? Now, bear in mind that I had promised you guys at the start of the semester that I'm dropping the lowest grade, right? So if you didn't do too good in this one, no sweat, just make sure that you do good in the others. Okay? All right, let's continue our class. Have you guys seen this diagram before?
Yes, sir.
Okay, so we have discussed this diagram in class, right? And I think I showed you guys this video on DNA replication. Did I? All right. So now I think we'll continue from here, which is transcription. So remember, the central dogma of molecular biology is DNA replication. And bear in mind that when we talked about DNA replication, that entire DNA molecule replicates. The entire DNA molecule makes a copy of itself to pass to the progeny cells. And that happens every time you have mitosis. So every time you have mitosis and cells are dividing, that DNA must make a copy to pass a copy to the progeny cells. So now we move on to transcription, whereby portions of that DNA molecule will be transcribed and produce a messenger RNA. So it is not the entire DNA molecule that will be transcribed. There are portions of that DNA molecule that actually codes for nothing. And so no transcription takes place there. Those portions of that DNA molecule that are transcribed are called what? So the portions of that DNA molecule that are transcribed are called what? I mentioned this the very first day of class.
By chance, is it genome? I'm guessing.
The genome, you said?
Yes, but I'm not certain.
The genome would refer to the complete genetic material of the organism. So the genome would consist of, for example, in the case of human beings, the genome would comprise of all 46 chromosomes. My question is, here you have a DNA molecule. All right, double-stranded. We said that when replication takes place, that entire DNA molecule replicates. It makes a copy of itself. However, transcription does not take place on that entire DNA molecule. You'd have transcription taking place, for example, here. This portion has been transcribed. Then somewhere over here, another portion has been transcribed. And everywhere where transcription occurs, it produces a messenger RNA. All right, single-stranded messenger RNA being produced everywhere where transcription takes place. So maybe transcription takes place here again. And it produces a messenger RNA. So back to my question. Those areas where transcription takes place, what do you call them?
The replication fork?
The replication fork, no. My question is, you see this portion right here? On that DNA that is in red, what do you call that? What do you call the portions on that DNA molecule where transcription takes place?
Sir, isn't that a non-coding strand?
No. Lourdes give up, and I was about to give the person 5 points to the next evaluation. 5 points towards the next evaluation. What do you call that portion on that DNA molecule where transcription takes place? I can clearly recall explaining this the first day of class. Not RNA sequence, not template strand, not RNA. I hope you guys are seeing my diagram. So you see right where transcription takes place, right here. All right, between this portion and this portion. So everything in here on that DNA molecule that you see I'm outlining in black, what do you call that? Remember that section on the DNA molecule that is transcribable produce a messenger RNA that will go to the ribosome for translation, and it will eventually produce a protein. No more guesses? Go ahead and write whatever you want in the... No, it's not the coding strand, not the nucleus. It's not single-strand RNA. What do you call that portion on that DNA molecule where transcription takes place? All right, it seems like nobody's interested in my five points that I was willing to give away. So the answer is gene. That portion where transcription takes place is a gene. All right? We said that human beings have approximately 25,000 genes on the DNA molecules. All right? And these genes are distributed along all 46 chromosomes. However, there are some regions of this DNA molecule that does not transcribe because they don't code for anything. All right? They don't code for anything. So all these 25,000 genes that we have are distributed along all 46 chromosomes that we have. All right? So remember that you have a one gene, one enzyme hypothesis, whereby each gene is responsible to produce a protein. And sometimes that protein is a structural protein, is a functional protein, or it could even be an enzyme. All right? Remember that in the particular case of albinism, albinism results from a defective gene that is responsible to produce an enzyme called tyrosinase. So tyrosinase is an enzyme, and when the gene that is responsible to produce that enzyme is defective, you will end up with albinism because that enzyme has a key role in the production of melanin. All right, so when you have a defect in that particular gene, which is responsible to produce that enzyme, then you can end up with these genetic conditions like albinism. All right. So let's get into more details about transcription, right? They say transcription is a transfer of information from a double-stranded template DNA molecule to a single-stranded RNA molecule. So basically, it is to get the information from that DNA molecule, and that information is stored in that single-stranded RNA molecule, which will take to the ribosome and tell the ribosome which protein or enzyme to produce. Okay, so that is transcription. Translation, which is protein synthesis, is the conversion in the cell, particularly in the ribosome, of that messenger RNA base sequence information into the amino acid sequence of a polypeptide. So the role of the messenger RNA is to take a message, a coded message from that DNA to the ribosome, telling it which protein to make or which enzyme to make, because remember that enzymes are protein by nature. Okay, so that is translation. But we will focus our attention first on transcription. All right, we will focus on transcription. All base pairs in the genome are replicated during the DNA synthesis phase of the cell cycle. But only some of the base pairs are transcribed into RNA. That is what I explained in my diagram drawing that DNA. All right, the DNA molecule. Only some of those base pairs will be transcribed into RNA. The specific sequence of base pairs that are transcribed are called genes. And thus the transcription process is also referred to as gene expression. So when we talk about gene expression, all right, it's extremely important, especially in genetic engineering, because you can have a portion there which is actually a gene. It actually codes for something. But because of the developmental stage in that particular organism, it does not transcribe. So no messenger RNA is being produced. So in order to determine whether your gene is being expressed or not, you can find out if there are messenger RNA. If messenger RNA is being produced, then you can safely say that that gene is being expressed. If no messenger RNA is being produced, then that gene is there, it is dormant, and it is not being transcribed. Now, I want you to take focus on this. There are times when genes are present, but they are not being expressed because of the developmental stage of that particular organism. So you have organisms that developed, right? And you have different stages. And there are times when certain genes are there, but they're not expressed until it is the right time in the developmental stage of that organism. All right? So back to what I'm saying about gene expression. How can you know whether a gene has been expressed or not? If there is presence or not of messenger RNA. And you can even go further to determine whether there are presence or not of that particular protein. So once there is no protein or the enzyme, or there is no messenger RNA for that particular sequence, then that gene is not being expressed. All right, so the transcription process sometimes is referred to as gene expression. Okay. Around the beginning and end of each gene are base pair sequences called gene regulatory elements, which are involved in the regulation of gene expression. These regulatory elements are referred to at the start would be promoter, and at the end, terminator. All right, so we will see in just a few the details of transcription. The promoter must be recognized first because that is the start. There is where transcription will begin. So it is important that the promoter be recognized. That way, all the components responsible for transcription know exactly where transcription should start. And of course, the terminator, where it should end. All right, so here we have chromosomes. This is a chromosome, and along this chromosome, you have genes, different genes. All right, so it says genes, segments of DNA that code for specific traits. So you can have gene for height, gene for eye color, gene for everything, right? Gene for skin color. But like I said, there are certain portions on that DNA molecule that don't code for anything. All right? So if we look at this diagram here, which summarizes transcription, what you see there in that blue, that double-stranded molecule, that is the DNA. All right? And so right there, the first thing that must take place is the denaturation at that portion right there. So DNA must unwind, right? It must be denatured and expose the bases. So remember, these bases here are exposed. All right, these bases are exposed. And I want you to pay keen attention to what happens. Only one strand of that DNA molecule will be used to produce a messenger RNA. I repeat that. Only one strand of that DNA molecule will be used as template to produce that messenger RNA. And that strand is called a template strand. So this is the strand right here at the bottom that is used to produce that messenger RNA that you see there in red. So what you see there in red is the messenger RNA being produced during transcription. The other strand, the one at the top, is referred to as the coding strand or the non-template strand. A lot of students get confused with this when they hear coding strand. They think the coding strand is the template strand, all right? And so they would build a messenger RNA from that coding strand. And in fact, that's an error. So the other strand that is not used to produce the messenger RNA is called the coding strand or the non-template strand. The enzyme that catalyzes this particular reaction is called the RNA polymerase. All right, so that's the enzyme that catalyzes, that's the enzyme that will be bringing corresponding bases on this template strand in order to form that messenger RNA. Of course, after this messenger RNA has been built, that DNA molecule comes back together. It renatures. The DNA molecule renatures after the messenger RNA has been built. Okay? So here we have, these are the NTPs, the bases. We're talking about A, G, C, and U. Those are the NTPs that the RNA polymerase will use to match on those exposed bases on that template strand in order to build that messenger RNA. Okay? This region right here, where you have both RNA and DNA molecule, that is the RNA-DNA hybrid region. But eventually, that RNA molecule will come off from that template strand and go on its own towards the ribosome. Okay? So this summarizes transcription, all right? But we will have to look at transcription in more details in prokaryotes as well as eukaryotes because there are some differences there. There are 4 major classes of RNA molecules or transcripts that are produced by transcription, all right? 4 major classes of RNA. Of course, there are many other RNA molecules, but these are 4 major classes that we will discuss. The first is the messenger RNA, small r, capital RNA, messenger RNA. We all know what's the role of the messenger RNA, right? Take that coded message from the DNA molecule towards the ribosome, and it tells the ribosome which protein it should make. Okay? Transfer RNA, small t, capital RNA, transfer RNA. The role of this transfer RNA is to bring, so right there in the ribosome, when translation is taking place, this transfer RNA is the one responsible to bring the corresponding amino acids to build that polypeptide. All right? That's a transfer RNA. Ribosomal RNA, small r, capital RNA. These form a part of the ribosome. So these help form the ribosome, which is that organelle where translation takes place. And then you have small nuclear RNA, small SN, capital RNA, small nuclear RNA. This plays a key role only in eukaryotes, and its role has to do with the final processing of that messenger RNA. We will note that I mentioned that transcription is different in prokaryotes than in eukaryotes. It says here the first 3 are found, so the first 3 from 1 to 3 are found in both prokaryotes and eukaryotes, while the last one, which is small nuclear RNA, are only found in eukaryotes. This is because the messenger RNA in eukaryotes require further processing before translation can start. And during that process of processing, this small nuclear RNA plays a key role. All right? I'll explain that in more details when we reach there. All right? So take note. Small nuclear RNA is only found in eukaryotes because the messenger RNA in eukaryotes requires further processing before translation can start. Okay? Only the messenger RNA molecule is translated to produce a protein molecule. So a gene that codes for a messenger RNA molecule and hence for a protein is called a structural gene or a protein coding gene. All right? Transcription is catalyzed by an enzyme called RNA polymerase. The DNA double helix must unwind, all right? So it must denature, open up before transcription can begin. Only one of the two DNA strands is transcribed into an RNA. This strand is called a template strand, and the other strand is called a non-template strand or the coding strand. Okay? In transcription, the RNA precursors are the ribonucleoside triphosphates, which would be ATP, GTP, CTP, and UTP. Remember that the U is exchanged, the T in DNA is changed to the U, uracil, right? In RNA. So collectively, all four of these ribonucleoside triphosphates are called NTPs. So those NTPs are needed for transcription. Those are the bases, the nitrogenous bases that will be used to build that messenger RNA molecule. You should note that if I give a nucleotide sequence, for example, and I ask you, is that a DNA molecule or an RNA molecule? Right away from the presence of that U there, you should know that that is an RNA molecule. You don't say any T. All right, so there's no T in an RNA molecule because that T is swapped for U. All right, so thymine is swapped for uracil. So this, for example, would be an RNA molecule because of the presence of that uracil. RNA polymerization is very similar to DNA polymerization reaction. When we talk about polymerization, we're talking about the process of adding these bases in order to build that molecule. All right. So RNA polymerization is very similar to DNA polymerization reaction. The next nucleotide to be added to the chain is selected by the RNA polymerase for its ability to pair with the exposed base on the DNA template strand. So it is the RNA polymerase that will be bringing the corresponding NTP, all right, in order to build that messenger RNA. As in DNA replication, RNA is synthesized in the 5' to 3' direction. So always remember that. So DNA replicates in the 5' to 3' direction, and RNA is also built in that 5' to 3' direction. Okay? To initiate transcription of a gene, RNA polymerase binds to a transcription controlling sequence adjacent to the start of the gene. This sequence is called the promoter. All right, so each gene has at the start of it a promoter. And it's important that that promoter is there because it has to recognize where that promoter is in order to know exactly where transcription should begin. So transcription will always start at the promoter region and ends at the terminator. So besides RNA polymerase that plays a key role in recognizing where the promoter is, there is something called the sigma factor or the factor sigma. It doesn't matter how you say it. The sigma factor is essential for promoter recognition. If sigma is absent, the core enzyme initiates transcription randomly. So there are two things that play a key role in recognizing where that promoter is: RNA polymerase and the sigma factor. Two of these will find where that promoter is and know exactly where transcription should commence. All right? Transcription ceases when a controlling element, which is called the transcription terminator sequence, or a terminator, is encountered at the end of the gene. So you have RNA polymerase along with sigma factor recognizing where the promoter is. And right there at that promoter, it will start to denature that molecule and start to transcribe that portion. And transcription will run along that template strand of the DNA molecule until it encounters the terminator. And when it encounters the terminator, there is where transcription stops, and that RNA molecule is being released from the template strand. Okay? The RNA polymerase enzyme performs several functions. We mentioned two of them already. All right, we mentioned a couple of them. The first is that it recognizes a promoter on the double-stranded DNA. So that is one. That is the first function of that RNA polymerase. Recognize the promoter. Second, it causes the DNA to denature and unwind into single strands at the promoter, similar to the initiation of DNA replication. All right. Third, as a result of reading the promoter sequence, the RNA polymerase orients itself properly and transcribes the entire template strand of the gene, which means that the RNA polymerase is the one responsible to bring the corresponding NTPs, the corresponding bases, in order to build the messenger RNA. That is the role of the RNA polymerase as well. And lastly, it stops transcribing when it reaches and recognizes the terminator. So the RNA polymerase also recognizes where that terminator is and knows exactly where to stop transcription. Okay? So here you have a synopsis of what that gene is. So you have a promoter at the start. So this is a gene, right? The regions of a gene. So you have the promoter. So right here, that will be recognized by the sigma factor and RNA polymerase. And when that recognition occurs, it starts to denature that gene right there at the promoter. And then RNA polymerase helps to, of course, denature the molecule and then transcribe all along that RNA coding sequence until it encounters this terminator. So the RNA polymerase is also responsible to recognize where that terminator is, and there is where transcription will stop. So these are the main regions of a gene: promoter, the RNA coding sequence, and then the terminator. Okay? Transcription in prokaryotes and eukaryotes are similar. The main differences are that more than one RNA polymerase enzyme occurs in eukaryotes, and that the transcription sequences are different. When we talk about the transcription sequence, we mean the promoters and the terminators. All right? So the whole process starts with initiation, elongation, and then termination. Initiation has to do with the recognition of the promoter. Elongation has to do with the polymerization of that RNA molecule, and the termination has to do with recognizing that terminator and ends the transcription process. Okay? So in this diagram, we notice here that you have the promoter. So this is the initiation, right? So the initiation has to do with promoter recognition, and that is done by what? RNA polymerase and the factor sigma, or sigma factor, whichever one you want to call it. So that is initiation. So initiation has to do with promoter recognition by these two components: recognizing where that promoter is, and then that RNA polymerase will denature that double-stranded DNA molecule right there at the promoter. And of course, the same RNA molecule will be responsible to transcribe that RNA coding sequence. Okay? So if you notice, elongation has to do with building that RNA molecule. And that will continue until it encounters the terminator. And that is the last part, which is termination. Encountering the terminator, and there is where transcription will end. If you notice what will happen here, this will eventually renature, come back together. That double-stranded DNA molecule will eventually come back together. And then you have your single-stranded messenger RNA molecule here in yellow that will then move towards the ribosome for translation. Okay? So I mentioned this already, but these are the role of the three types of RNA. So the messenger RNA, copy of gene. So it is made in the nucleus in the case of eukaryotes and move out to the ribosome. It holds the code for amino acids to make proteins. All right. So the messenger RNA is a coded message from that DNA molecule, and it goes to the ribosome telling it which protein to make. The ribosomal RNA, small r, capital RNA, helps to form ribosomes. And then transfer RNA transfers appropriate amino acids to the ribosome for building the protein, the polypeptide molecule. The small nuclear RNA, we'll come to that in just a few when we discuss the details, the molecular details of transcription in eukaryotes. Okay. So basically what you have here is, this diagram is depicting what takes place in eukaryotes, because if you notice there is a nucleus. So remember that in prokaryotes there is no nucleus. So here you have the nucleus. All of this that you see here on the top left-hand corner, that's the nucleus. If you notice the pores, right? So those are pores on the nucleus. And what do you see coming out of the pore? This right here. This right here. All right. Which is the messenger RNA. That is the messenger RNA. And where does it go? Towards the ribosome. So this right here is the ribosome. This organelle right here is the ribosome. All right. And of course, ribosomal RNA helps to form that ribosome. And the role of the transfer RNA is to bring corresponding amino acids to the ribosome in order to build the polypeptide. This what we see here, these three amino acids, that is the polypeptide being formed right there. So remember that the polypeptide is a sequence of amino acids, right? That's a protein molecule. It's a sequence of amino acids. How does it know which amino acid to bring? Well, it is based on the codon that we have here. All right? Based on the code that is on the messenger RNA. All right? And so you have the anticodon from each transfer RNA. All right? And that brings the corresponding amino acid to the ribosome in order to build the polypeptide. Okay? Here is where there's a huge difference between transcription in prokaryotes and that of eukaryotes. But we will stop there for right now because this requires some explanation. And it's very important that you understand this major difference in transcription between prokaryotes and eukaryotes. You need to understand that. And there is where we'll understand the role of the small nuclear RNA. All right? So we'll stop there for today and then we'll continue this discussion on Thursday. Any questions before we leave?